So thank you very much, Carla, for the invitation to be here today. It's a real honor to be here. I mean, I think for many of us women, biologists and biological engineers, Rosalind Franklin is a true hero for us, or heroine, I suppose. So I thought I'd just use today to talk a little bit about what I do and what gets me excited and what attracted me to science and engineering. For those of you who don't know, my name is Reshma Shetty. I'm a co-founder of a company in Boston called Ginkgo Bioworks. If you're ever in Boston, you're welcome to come by for a visit. We are a synthetic biology company. We design organisms, and that's what we're excited about. I oftentimes like to start my talk with this slide, particularly if it's sort of a mixed audience. Does anyone know what the most advanced piece of technology on this desk is? Huh? Anybody shout it out? The plant? That's right. Well, yeah. Rosalind Franklin Society. Trust you guys to get it. In contrast to sort of every other piece of technology on this, the only piece of technology on this desk that can self-repair, self-assemble, self-replicate, and is renewable is the plant, right? And I think, even as biologists and biological engineers, we oftentimes take this for granted, right? The dying houseplant in the corner of your house has these innate capabilities that every other engineering discipline can only dream of, right? The electrical engineers wish they could do this, right? And so what fascinated me when I first got to MIT back in 2002 was that, my gosh, if you could figure out how to harness these capabilities from biology, the sky's the limit in terms of the applications, right? And I know I'm preaching to the choir here, particularly after Susan's talk, but I think truly biology is the most powerful manufacturing platform there is. The challenge is we just don't know quite how to fully harness it and really how to use it. I think the other thing that fascinates me about biology and what attracted me to this field in the first place is that at its core, biology is digital, right? The A's, P's, G's, and C's of an organism, the genome of an organism, dictates its behavior. And if I can figure out how to rewrite those A's, P's, G's, and C's, I can elicit a new phenotype, a new behavior from that organism, right? Now, the problem is that we're not very good at knowing how to program biology, how to recode biology. And so we started Ginkgo really with the mission of making this process of designing biology, of engineering biology, faster, easier, and cheaper. And that's what we do and think about day in and day out at Ginkgo. Now, Susan already showed you a couple of these trends, right? The dotted line there is the rate of improvement in computing technologies, right? It's Moore's law. It's saying that the density of transistors on a silicon wafer it is getting more and more dense every 18 to 24 months. And she also showed you the second line, that DNA sequencing, the cost for sequencing a genome has fallen dramatically. Most people are aware of both of these trends. I think the trend, the third plot is the one I actually want to draw your attention to, which is this one here. This is the cost of DNA synthesis, right? So DNA synthesis is the writing of DNA. And specifically, this is gene-length DNA synthesis. So if you think about it, right now we are very fortunate to be living in a time where the cost of reading DNA has fallen dramatically, and the cost of writing DNA is also falling pretty dramatically. And if you can read DNA, if you can write DNA, then you can start to think about how to actually be able to program biology, right? And so at Ginkgo, as well as in the synthetic biology field in general, we are riding two very powerful, exponentially improving technology trends to be able to improve our ability to engineer organisms. Now, there's a third trend, or I suppose a fourth trend that's not listed on this slide, which is improvements in measurement technologies. And so, if you're familiar with mass spectrometry technologies, they're also getting better year after year. You can go buy a new instrument from Thermo and it can measure things with a greater degree of sensitivity and precision than it could the year before. So combined, these reading DNA technologies, writing DNA technologies, and measurement of cell technologies together have sort of converged and have greatly unlocked our potential and our ability to engineer biology, to program cells. So at Ginkgo, we think a lot about this, right? Basically, we're genetic engineers. And so we think about how good are we at programming cells. And so this is a plot of basically the number of unit operations. You can essentially think of these as genetic engineering unit operations that are happening at Ginkgo per month. And you can see this is the data we've collected from, this is going from 2015 to 2018 and into 2019. And you can see that basically we've been increasing the number of unit operations per month, again, at an exponential rate, right? This is a log scale here. Now, we have not been growing our team size exponentially during that time. And so what that means is that we're actually also getting better at doing genetic engineering of organisms every year at Ginkgo. And if you get better at engineering organisms, then new applications open up, right? So I wanted to just talk about a few of the types of areas where we're trying to deploy these biological technologies. So we sort of think of ourselves as a platform company. We sort of think of it as we're building the back end of the bioeconomy. And the interesting thing about biology is it touches all facets of our lives, right? Ourselves, our environment, our food, our health, they're all connected to biology in one way or another, right? So again, if you can unlock our ability to engineer biology, which to be clear is a very hard problem, right? Biology is still really complicated. We're still, frankly, just only scratching at the surface of our ability to engineer it. But if you can get better at it, then lots and lots of application areas end up opening up. So one project we have that I'm super excited about is we started a joint venture with Bayer Crop Sciences. And Bayer recently acquired Monsanto, which you probably are more familiar with. And so Bayer is the largest ag company in the world. And a couple of years ago now, they came to us and said, hey, you know what? We really want to see if we can take a swing at some really big moonshot problems. And specifically, they were interested in nitrogen fixation. So, about 100 years ago now, we invented the Haber-Bosch process, right? The Haber-Bosch process allows us to make nitrogen fertilizer. And that was basically what led to the population explosion we have today, right? The only reason we can support this growing population on Earth is because we can grow food with much higher yields than we could before Haber-Bosch, right? And so it's driven a huge amount of improvement in terms of our food system, but it's also an incredibly energy intensive process, right? And it also contributes a ton to climate change and whatnot. And so if we could figure out a way to fix nitrogen, not using chemistry, but using biology, that would be huge. Now, it turns out there are microbes that already do this, right? There are plants that can essentially don't require as much fertilizer because there are microbes living in and around the roots or even inside the plant that can fix nitrogen for the plant. This is not without precedent. The problem is that just like the chemical process, the biological process is energy intensive. And so these microbes tend to shut it down as soon as there's any fixed nitrogen around in the form of fertilizer. So if we can figure out how to get these microbes to keep biological nitrogen fixation pathways on, even in the presence of fixed nitrogen, you can start to reduce chemical fertilizer usage and supplement it with biological nitrogen fixation. And so this is very much in the early stages, still a development project at Join, but one, if we can actually figure it out, has the potential to have a huge impact on the world. And so it's one I'm excited to take a swing at, even if we're not successful. We have a similar effort going on in a new company that we spun out called Motif Foodworks. Motif is really interested in the food space. And again, our food supply has a pretty significant environmental footprint in the world, right? And so there are a whole set of companies out there now trying to take a swing at could we build a more sustainable food supply. And specifically, have folks heard of the Impossible Burger? Anybody tried it? Yeah, woo! So what makes the Impossible Burger actually pretty tasty for a veggie burger? It has a protein in there called heme. And Impossible makes that protein using engineered yeast, right? So they've engineered yeast to produce this heme protein. They grow it up in tanks, very similar to brewing beer, purify the protein, and include it in their Impossible burger. And so the goal is to go to a more essentially sustainable food supply. And so inspired by Impossible, we actually spun out a company, Motif, to basically look at what other proteins can we make using fermentation. Could we make milk proteins? Could we make egg proteins? Could we start to basically transform how we go about making animal products, right? Instead of, sourcing them from animals, can we source the key proteins from fermentation and then use plant-derived protein sources to make these food products? And so we're really excited about this and about the constellation of protein ingredients that we might be able to bring out. We also partnered with a company called Genomatica. Now, Genomatica has been around for a while now. They're a company in San Diego. They're really interested in the chemical problem, right? The fact is that most of our chemicals and most of the plastics that we have, like pretty much, that chair, the carpet, the paint on the walls, all of this comes from a barrel of oil, right? And so if we want to move our chemical production to a more sustainable footprint, we need to think about ways to have biological production of these chemicals instead. And so Genomatica actually was able to develop a process for making what's called BDO, butanediol. And this is basically a chemical precursor that can go and basically you can formulate into plastics. So they have a scaled up process operating in Italy right now, and are basically selling into the European market because there's been a much bigger driver for renewable plastics in Europe than there is in this country. And so you can go to the grocery store and your plastic bags are made from Genomatica's BDO process. And so we're really excited to be partnering with them to go after additional chemicals and try to transform towards bio-based production. About a year ago now, we ended up acquiring a company called Warp Drive. Warp Drive was taking a really interesting hack at a very tough problem, which is that I think probably a lot of folks here have heard about the rise of multi-drug resistant microbes, right? We're starting to basically run out of antibiotics against certain types of infections. And so what Warp Drive did is they said, well, microbes are really good at fighting other microbes. What if we go and start sequencing interesting families of microbes, basically sequence the dirt, and find interesting new chemistries, interesting new potential antibiotics. And so they did that. They sequenced about, do I have it here? No, I don't have it here. I think it was about over 100,000 different microbial genomes, right? And started using computational tools to sort of predict what interesting structures might be buried in this genomic content. And so we're working with Roche to basically identify interesting chemical entities and screen them for antibiotic activity in the hopes that we can bring new antibiotics to market ultimately. And so really excited by this genome mining platform that Warp Drive's developed and that we've now integrated into the Ginkgo platform. And then finally, I'll just end with talking a little bit about a recent partnership that we have with Synlogic. So Synlogic's a company in Cambridge, Massachusetts, who's really interested in developing living medicines for things like inborn errors in metabolism, right? So, there are folks out there where when they're born, they're missing a metabolic enzyme. That means they start accumulating certain metabolites in their body. So, for example, in particular, we were focused on a disease called maple syrup urine disorder. This is where people start accumulating branched chain amino acids, right? Because they don't have the metabolic enzymes to basically clear these out of their system. And so Synlogic's hypothesis was like, hey, maybe we could engineer a microbe to consume leucine for patients afflicted with inborn errors in metabolism. And so they worked on this and developed a proof of concept, but their proof of concept didn't work well enough. So it would clear leucine in sort of in an in vitro test tube model, but not actually in a mouse. So we collaborated and worked together to basically take Ginkgo's platform and try to improve their living medicine. See if we could actually make it be able to clear leucine from an animal model faster. And so we went through 2 rounds of running our platform, basically designing a new microbe, building it, testing it. And at Ginkgo, we tend to do everything in high throughput. So when I said we did this actually a couple times across thousands of different designs in each cycle. And at the end, we were able to come up with an improved microbial candidate. That's the SYN 5941. And we actually ran an experiment looking at leucine clearance in a primate model. And so you can see here, this is the vehicle. This is Synlogic's original prototype strain, which basically doesn't clear leucine from the animal model, whereas this improved microbe that Synlogic and Ginkgo were able to work together does actually clear leucine at significantly improved rates. And so obviously lots more work to do on this before it would be ready for clinical trials in prime time, but promising to show how Ginkgo's platform combined with Synlogic's expertise around these inborn errors in metabolism has the potential to develop whole new therapeutic modalities, right? Where your drug is not a small molecule. It's not even a biologic. It's actually a bacterial therapeutic that you could take to help clear some of these metabolites from your system. And so with that, I'll wrap it and say thank you again for the invitation to be here. It's really an honor.